In the last part of this chapter, I want to talk about the cell junction or membrane junction and also cell organelles. We have three types of junctions between the cells. One of these junctions is called tight junction. Tight junction, as you see here, are the strands or rods of protein which can make link between two cells. As you see here, the tight junction is the first junction from the apical border of the cell. If this is the apical border, this is the basal border, and here is the lateral border between the cells. The tight junction is the first one which you can see in the near the apical border. This junction prevents substances from passing between cells, so it doesn't let anything to pass between the cells. The other type of junction between the cells is called desmosome. Here you can see desmosome between two adjacent cells. In the desmosome, you can see a protein plug in the inner leaflet of each cell, as you see in this picture. And then we have some filaments which are attached to these protein plug. And these proteins invaginate to the space between the cells and connect the cells with each other. In the desmosome, we have a distance between two adjacent cells, which is about 20 nanometer. And the other junction between the cells, which is located near the basal border of the cell, is called gap junction. In the gap junction, you can see tiny fluid-filled tunnels between two adjacent cells. Between these cells, you can see the tunnels which can help material to pass from first cell to the next one. We have some ions, we have ATP, we have electrical signals which can easily pass through the gap junctions. These tunnels are made up of integral proteins which we call them connexon. So these are the junctions which we have between adjacent cells. First, I want to talk about the membrane-bound organelles. The membrane-bound organelles are the organelles which are enclosed with the membrane. And they allow activity in isolated environment. One of them is the endoplasmic reticulum. The endoplasmic reticulum, as you see in this picture, these are endoplasmic reticulum. And the endoplasmic reticulum are extensive interconnected membrane network. They are varies in their shape, but all of them are continuous with each other. Extend from the nuclear envelope, as you see here, to the plasma membrane of the cell. And they compose about half of the membranes within the cell. We have two types of endoplasmic reticulum. One type of this endoplasmic reticulum is called rough endoplasmic reticulum, which you can see it here. And here you can see the functions of rough endoplasmic reticulum. They contain ribosomes on their surface. Because of their ribosomes, we call them rough. They can synthesize protein for secretion, and it can make proteins which we have in the cell membrane and enzymes which are located in the lysozyme. They can process molecules, they can modify protein, add carbohydrate to the protein to make glycoprotein, and they can tag them for shipping, and it can store protein for us. The RER or rough endoplasmic reticulum can help formation of some organelles like peroxisomes, and they can make vesicles. They can form transport vesicles for shipping of protein to Golgi Apparatus. The other types of endoplasmic reticulum is called a smooth endoplasmic reticulum. As you see here, we don't have any ribosomes on the surface of a smooth endoplasmic reticulum. They can synthesize lipids, They can process some molecules like carbohydrate. They can do detoxification of drugs and poisons. They can make vesicles, the transport vesicles, which can bring these carbohydrates to Golgi apparatus. And also, one of the important function of them, which we don't have it in this list, is a storage of calcium ion, specifically in skeletal muscles. The next organelle which we have in the cell is called Golgi apparatus. This is the structure of Golgi apparatus. As you see here, the Golgi apparatus are composed of these flattened sac, elongated sac membrane-bound structures. And they have two surfaces. One surface, which is toward the smooth endoplasmic reticulum, is called cis face, and the other one, which is toward the cell membrane, is called trans face. Here you can see cis face, and here you can see the trans face. Their function is modification, package, and sorting of protein. They are like the post office of the cell. They can get the protein and carbohydrates and lipids. They can add these material on each other, pack them, sort them, and prepare them to go out of the cell and do exocytosis. So formation of secretory vesicles, which you can see here, is one of the functions which we have for the Golgi apparatus. The other structure, the other organelle which we have in the cell is called peroxisomes. The peroxisomes are small membranous sacs. They contain digestive enzyme in the cell, and these digestive enzyme and the lysosome are formed by Golgi apparatus, and the enzyme are formed and made by the rough endoplasmic reticulum. They are important for digestion of unneeded substances, specifically pathogens which come to the cell, and sometimes our cells want to do suicide. They want to kill themselves, which we call them autophagia. So this lysosome start to release their content inside the cell, and by digestive enzyme they can solve everything inside, and they can do autolysis or killing a cell. The other structure organelle which we have in the cell is called peroxisomes. Peroxisomes are membrane enclosed organelles. They are very small sac, smaller than lysosome. They pinch off from the rough endoplasmic reticulum, and they have different types of proteins, which most of them work as enzyme. And they have some functions like chemical digestion. They can do oxidation, and also they can make lipid. So lipid synthesis is one of the function of peroxisomes. We have some endomembrane system in our cell. These endomembrane system are extensive array of membrane-bound structure, include endoplasmic reticulum, Golgi apparatus, vesicles, lysosomes, and peroxisomes. They include plasma membrane and nuclear envelope. They start from nuclear envelope and continue to the plasma membrane. They connected directly or through vesicles which move between them, and they can provide means of transporting substances within the cells. The other organelle which we have in the cell is called mitochondria. The mitochondria is the powerhouse of the cell. The mitochondria is elongated shape organelles, and it contains two membranes, one outer membrane and one inner membrane, which contain many folds. And the inner membrane is called crista. And inside the mitochondria, we have a matrix. and by using oxygen, they can do cellular respiration and making ATP. We call them powerhouse of the cell. And we have a group of non-membrane bound organelles. One of them is ribosomes. Ribosomes contain protein and RNA. They are arranged into large and small subunits. Here you can see the picture of the ribosomes. This is the large subunit and this is the small, and they are located on each other, and the protein is made between them. And we have two types of ribosomes. One type of ribosome is called bound ribosomes or fixed ribosomes. They are attached to the external surface of the endoplasmic reticulum. They are important for making protein for export. And the other type ribosome is called free ribosome, which are located inside the cell, and all other protein which we need them for the cell are made in the free ribosomes. The other structure is called centrosome or centrioles. They are pair of perpendicularly oriented cylindrical centrioles. As you see here, they are located on each other perpendicular, and they are surrounded by amorphous protein. Their primary function is they can organize microtubules, which we have them in the cytoskeleton of the cell. They are important for cellular division, and they can make the flagella in the sperm. The other organelle which we have in the cell is called proteasome. The proteasome are large barrel-shaped protein, as you see here. They are protein digesting organelles. They are located in cytosol and also in the cell nucleus. And they can break down the cell proteins by using ATP. And as you see here, the protein come inside this barrel, and the amino acids and the peptide fragments come out from the other way. And proteins their function, one of the important function of them is a protein digestion, the protein which our cells doesn't need them. With age, may be unable to normally remove protein for us. The other non-bound membrane organelles are cytoskeleton. They play a role in the intracellular support, organization of the organelles, cell division, and movement of organelle. And they are located in all parts of internal of the cells, and they attach to the anchoring protein which we have in the cell membrane. They are divided into three groups. One of them is called microfilament. One of them is called intermediate filament, and the last one is called microtubules. Here you can see these different shapes of cytoskeleton. You can see microfilament here. The microfilaments are the smallest component of the cytoskeleton. In the microfilament, we have actin protein, which are twisted filaments, and their function is maintaining the shape of the cell. They can support the structure of the cell, and they are important in cell division because they can make the spindle of our division. Some of them are called intermediate filament. The intermediate filament are intermediate in their size. They are more rigid than microfilament, and they can do a structural support and cell junction. And some of them are called microtubules. They are the largest component of cytoskeleton, and they contain a protein which we call it tubulin. And they are very important for making the shape of the cell, cell division, and cell transport. The material wants to move in our cells are moved by these microtubules. And we have some structures which are located on the surface of the cells. I talked about cilia before for you. They are hair-like projections which can move forward and backward and sweep every particle which we have on the surface of our cells. We have flagella which are longer and wider than cilia, and proper entire cell like the flagella which we have in the sperm, and also we have microvilli. The microvilli are extension of plasma membrane which can increase surface of absorption. And in this picture you can see these microvilli which are the finger-like projections on the surface of our cells. And now you can answer these questions. You can describe the general structure of both endoplasmic reticulum and Golgi apparatus and their functional relationship in synthesis, modification, storage, and release of molecule. Lysosomes and peroxisomes are both a small membrane enclosed sac. Which of these function is both digestion and synthesis of molecule and digestion of unwanted organelles? Which non-membrane bound organelle functions can digest unwanted material? Which one can form the structural support of cell? Which one can make protein? And which one can participate in cell division? And which cellular surface structure can increase the surface area of the cell, and which one can move material past the cell, and which cellular junction can provide resistance to mechanical stress, which one allow passage of ions between the cells, and which one prevent leakage between the cells. We are done with the cell biology. In the lab part, we may talk about the mitosis and meiosis division, and you will be more familiar with the structure and function of the nucleus. Let me know if you have any other questions in this part.